starting again this time. I'm Stephan, and my project this summer was characterizing the immunological inflammatory response after a cell is infected with both MHV68 and LPS. So just some back information about pregnancy. During pregnancy, there are a number of immunological and physiological changes that occur to support the fetus. The placenta here is of important nature. As it is derived by the fetus, it provides essential nutrients and oxygen to it and removes waste products from it. The maternal side of the placenta is called a decidua. It protects the fetus from a maternal immune rejection as well as infections. And on the right shown here is the maternal fetal interface. There are a lot of immune cells such as T cells and macrophages, and it is also mainly composed of placental or trophoblast cells as well as decidual stromal cells. So viral infection. After someone is exposed to a viral infection, a subsequent bacterial infection will happen, and then inflammatory response will be produced. Inflammation is when a lot of immune cells are being recruited to the site of infection, producing pro-inflammatory cytokines to exacerbate the immune response. During viral infection, it's characterized when yeah, the camera. It's okay. It's okay. It's okay. During viral infection is when a virus is successfully reproducing inside of its host, causing a sequential inflammatory response. Now, during pregnancy, excessive inflammation can be dangerous to both fetus and mother. For example, it can cause preterm labor, preeclampsia, and for the child, it could cause neurodevelopmental issues, and the figure here displays such. So the adverse effects I just mentioned were manifested in the Spanish flu pandemic of 1918. Pregnant women, when exposed to a viral infection, were more susceptible to bacterial infection than normal pregnant women. Mortality rates amongst pregnant women with bacterial infection were abnormally high. And then also developmental issues for the fetus were manifested. However, there was no viral RNA detected on the fetal side, which suggests that the inflammation was crossing the placental border and then infecting the fetus. So in my project, I'll be characterizing this inflammatory response in hopes that future research could create a drug that would inhibit these detrimental excessive inflammation effects. So to do this, we use two entities, MHV68 and LPS. MHV68 stands for murine gamma herpes virus 68. It is a naturally occurring rodent pathogen, and in my project, it will be acting as a bacterial infection for our cells. A common property of this virus is to modulate the immune response, which is also present in our experiment results. LPS, also known as lipopolysaccharides or endotoxins, are composed of O antigen and gram-negative bacteria, and LPS is known to induce an immune response. So collectively, these two entities can effectively model what happens in a bacterial infection and subsequent inflammatory response. So type 1 interferons, oh, my bad, let's skip this slide. Oh, wait. No, this is going forwards. Okay, there. So LPS best responds to TLR4. After LPS binds to TLR4, it can induce two downstream pathways, one of which is the MYD88 dependent pathway, which would then induce inflammation such as TNF alpha, IL-6, IL-12 alpha. And the other side is the MYD88 independent pathway, which would then induce type 1 interferons. So type 1 interferons are key antiviral interferons. They would then induce ISGs or interferon signaling genes that would then produce pro-inflammatory proteins that would initiate the innate immune response. So our hypothesis, MHV infection during pregnancy leads to susceptibility to bacterial infection and a subsequent inflammatory response. So in order to prove our hypothesis, we had two cell lines, our trophoblast or swan cells, as well as our decidual stromal cells, also known as husky cells. Our four sample types are as follows: wild type plus saline, wild type plus LPS, MHV68 plus saline, and MHV68 plus LPS. In order to come to a conclusion, we use a lot of qPCR to have our results. Our main aim: does MHV68 modulate the LPS-induced inflammatory response at the maternal fetal interface? So we will first be looking at toll-like receptors or TLRs. So looking at our decidual cells first, we see that for TLR3, there is a decrease in the regulation for this molecule comparing wild type plus LPS and MHB68 plus LPS, whereas for TLR4, we see a relatively no change in the expression of this molecule. For our trophoblast cells, we see that there is a decrease in the expression of TLR3 comparing wild type plus LPS and MHB68 plus LPS, and the same results follow for TLR4. So what does this tell us? In trophoblast cells, we see the downregulation and transcription of TLR3 and 4, and in the decidual cells, we see there's no change in its regulation of TLR4. This suggests that MHB68 is preferentially modulating the immune response in these decidual cells. So we will now be looking at interferons. So firstly, for the decidual cells, we see that for interferon alpha, there's an increase in the expression for this molecule. However, for interferon beta, we see a decrease in the expression comparing wild type plus LPS and MHB68 plus LPS. The data for interferon beta suggests that MHB68 is indeed modulating the immune response. Now, from this, we can conclude that it is not modulating all type 1 interferons, rather just interferon beta. Looking at interferon lambda, a type 3 interferon, we also see that modulation occurring, which makes us hypothesize that is also modulated through interferon lambda. And then for our trophoblast cells, we see that for interferon alpha, there's a relatively no change. And for interferon beta, we further see the modulation occurring, and no results were detected for interferon lambda in our swan cells due to a high CQ value in our qPCR. So in summary, MHB68 suppresses in both cell types. However, MHB68 differentially modulates interferon alpha. We will now be looking at ISGs. So for our decidual cells, for MIX1, as we know, this is a molecule that is induced by interferon beta. So seeing the decrease in expression for this entity, we can validate our results for interferon beta. We do indeed see the decrease comparing wild type plus LPS and MHB68 plus LPS. For ISG15 and ISG20, we see either a no change or an increase. And knowing that these two types of ISGs can stem from type 1 interferons is not something that is surprising. And then for our swan cells, for MIX1, we can further validate our data from interferon beta, knowing that there is a decrease in expression for MIX1. And for ISG15 and ISG20, we also see the decrease in expression for these molecules. So takeaway. The effect of the suppression of interferon beta is manifested with the ISGs that interferons induce in both cell types. In other words, with a decrease in expression of interferon beta, it leads to a decrease in the expression of MIX1. And now we'll be looking at inflammation. So for our decidual cells first, we see an increase in inflammation now, comparing wild type plus LPS and MHB68 plus LPS. For IL-17 especially, we do see great increase in expression for these entities. The same thing follows through for IL-12 alpha, IL-6, and TNF alpha. For the swan cells, we see a decrease in induction of inflammation looking at TNF alpha, IL-6 especially, comparing wild type plus LPS and MHB68 plus LPS. And for IL-12 alpha and IL-17, we see a no change. So for going back to the decidual cells, we can hypothesize that the suppression of the MYD88 independent pathway leads to the hyperactivation of the MYD88 dependent pathway, inducing this inflammatory response. Inflammation will then only occur in the decidual stromal cells and not in the trophoblast cells. Our overall conclusion: MHB68 is preferentially modulating the immune response, and further, the inflammation stemming from the decidua and not the placenta. And I would take a few moments for acknowledgements. I would like to thank Anthony for your time and patience. I couldn't have done this with a lot of your help. And Dr. Moore, I'd like to thank you for this opportunity. It was unlike any other. And I also want to thank a lot of the faculty members, Dr. Albero, Dr. Ding, Dr. Tisha, Anna, Annie, Sandra, Yuan, Hussein, and Alex for all of your help in guiding me throughout my project this summer. Thank you. Any questions?
Ask me questions.
I already answered all my questions.
No questions?
Okay, very good. Thank you.
Thank you.